my great pleasure to give you some insight about this drug development. First, I'd like to thank to the invitation of this great meeting, KAT and also KoreaBio. So I'm an oncologist. I'm seeing the patient. I'm also doing the research. And also I'm the professor of the medical oncology and biomedical engineering at Emory University in Georgia Tech. So what I liked today, we give a little insight of the current drug development and what we're going to do in the future. I think this is a great forum to discuss about that. So this is showing nicely here. If you have, I think it's one of the big stone there. Do you know that stone? Anybody knows that? This is Stone Mountain. It is a single biggest stone in this planet. You know what? The height is about 250 meters, and then circumference is about 5 miles, 8 kilometers. It's a huge. There's a park there that is shown there. At Atlanta, we have two great universities, Georgia Tech and Emory University, and also the hub of the CDC, Center of Disease Control and Prevention, and also headquarters of the American Cancer Society is right there in downtown there. So Emory University consists of 7 different institutions. The first one is the Emory Main Hospital. So this is Main Hospital. This is Emory Midtown Hospital. And this is the VA Hospital. And this is Grady Memorial, St. Joseph, and Johns Creek, and Winship Cancer Institute, where I belong to. I think there's one interesting figure here. A couple of years ago, we have a pandemic of Ebola virus, right, in West Africa. And this actually, Dr. Kent Bradley actually worked there as a volunteer, and he got contracted the virus. He's almost dead. And he moved to Emory and get the treatment successfully. And there's about 3 or 4 very special units in the United States to treat such a patients, very well equipped. So Emory is one of them, and now he's a main center of the disease education and control there. But anyhow, today, this is my disclosure. I have research funding or advisory activity of these pharmaceutical companies and also the government funding here. So let's talk about mortality of disease. This is simply showing cardiovascular mortality and cancer mortality. So 1975, cardiovascular mortality is double than cancer. Cardiovascular mortality rapidly improved, but cancer mortality, not much. This is, of course, the statistics from the United States. But 1990s, at that time, it was gradually increased even, and then gradually decreased now. It's a long way to go to control this disease. So currently, cancer is the #1 killer in the United States. I think probably Korea may be similar too. So if you look at the cancer treatment history, you have surgery, which is the longest modality, still very important. Radiation therapy, chemotherapy, molecular target therapy. This is the last 10 years explode. And then nanotherapy now is going to go into, in my opinion. So this slide basically showing that evolving biomedical science. I think it's 1953, these 2 young men discover DNA double helix, right? You know that, who are they? Francis Crick and James Watson. James Watson, he's passed away, but James Watson is still alive. He's, I think, at the age of almost 90 right now. but he's still doing research. Then 2001, we have another breakthrough, which is completed Human Genome Project. After 13 years effort with huge amounts, $2.7 billion. Now, the science developed very rapidly, and now the same human genome sequencing actually can complete it within a day, and also very small money. The science is developed huge. And then at the same time, molecular target agents is being also developed very rapidly. So this is one of my contribution, just a few of them I just listed here. This regimen, paclitaxel phosphate and carboplatin combination, is actually developed by myself with my colleagues for neoadjuvant chemotherapy or recurrent metastatic head and neck cancer. And second another regimen is so-called cytoxan adiamycin cisplatin prednisone cap-prednisone regimen. This one still people using, but is a malignant thymoma, which is a very rare disease in your center of the chest. Biochemical prevention, we developed certain CRA developed heavily by the Ki-Hong, but adding interferon and vitamin E, we developed this one, chemopreventive agents, and that's also published. And then this cetuximab, I'm going to give you a little more detail of this. This is eventually got FDA approval. For the first phase 1 trial we conducted, and then phase 3 trials eventually get approval. So this one actually, initially, this compound developed by John Mendelsohn when he was in UC San Diego a long time ago, and then he became president of the MD Anderson. We worked together. But basically, this compound, probably everybody very well, you know that, human mouse chimerized antibody, and which is basically not growth inhibition of this growth pathway, but also, very importantly, antibody-dependent cell cytotoxicities are also associated with this compound. So this is our first human trial we conducted. Same time, single agent cetuximab. At that time, we called C225. Now we did patients this phase 1 trial before and after infusion. We did biopsy of the tissue, and then we did also cisplatin combination, and then the other institution also conducted C225 alone as a dose escalation at that time. Now we learned that this is very important. The baseline this one, 400 milligram per meter square for loading dose with 250 milligram per meter square maintenance dose weekly. Still we're using the same schedule. This very small study, only 12 patients entered, 9 patients evaluable. Look at the response rate is remarkable at the time. Of course, this is a very small study, but more importantly, we did this lady who had head and neck cancer, was a surgery and radiation therapy is done, and then recurrent disease, so-called dermal metastasis, which is also through the skin. This is a very nasty disease. She was treated with cisplatin 5-FU combination, but it didn't work. And then come to me at that time. This is one of my patients, and I treated with this compound, 400 milligram per meter square with a 250 milligram per meter square weekly. And then after 4 cycles of treatment, all these lesions cleared up completely. In fact, this lesion is still tiny bit there. So we did a biopsy this one, before and after treatment. And this EGFR expression before treatment quite high, but after treatment, about 80% reduction of this expression. You see the cells still there. So actually that dose, 400 milligram per meter square with 250 milligram per meter square, it worked very well, and there's still people using this schedule. So right after this, in fact, we did a major study embarked, phase 3, randomized trial with radiation therapy alone versus radiation plus cetuximab for locally advanced head and neck cancer, stage 3 or 4. This is was still standard treatment. And then the outcome, this is overall survival. This is combination with the cetuximab. And this one is a radiation therapy alone. Clearly improved survival. If you look at the median survival, 28 months versus 54 months. So based on that, actually FDA approved in 2006 for use of the head and neck cancer. At the same time, colorectal cancer and other disease also get approved. So what I try to show here, that this is a NCI initiative, National Cancer Institute initiative for genome studies. There's a several components. This is a very important study, and because of this, in fact, a lot of new drugs being developed. The Cancer Genome Atlas Project, or TCGA, and Cancer Stem Cell Project, Cancer Chemical Biology Discovery Centers, and the nanotechnology for drug development. And also, this is a little different, but comprehensive alternative medicine. And this one basically two major core functions in terms of tumor banking and bioinformatics. So this is basically all collaboration between the universities and industry. So let's give you a little explanation of the TCGA, which is embarked October 2006. Throughout the country, from the East Coast to West Coast, we have 7 different resources centers here. Biospecimen core, which is we have to collect all the human tissue, patient tissue throughout the country. Genome characterization centers, genome sequencing centers, proteomic characterization centers, data coordinating centers, cancer genome hub, and genome data analysis centers. This function is actually about almost 50 universities participated this. Of course, it's Hopkins and Emory and MD Anderson and many other institutions all participated in this trial. These are the studies. So this is one of the publication from that for lung adenocarcinoma. And this is actually published in Nature in 2014. If you look at their gene profile, you have KRAS mutation, EGFR mutation, BRAF, all other different mutations in adenocarcinoma in the lung we identified by this effort. And then of course we have targets for these mutations. So basically now not only lung cancer but also many other human cancers, based on this we are having target and then treated the patients. So you see now the old days, chemotherapy, lung cancer, such doublet, triplet, and then giving that era is gone. We don't treat it anymore. All the molecular targets and this particular mutation analysis is extremely important, and then also drug development through that, because of that, okay? So example, lung cancer here, ALK mutation, it's like a KRAS, is based on the ethnic group, there's a mutation rate is quite different. Caucasian about 30%, Asian is at 15%, but EGFR mutation different. Asian is higher, 40%, Caucasian about 15%. So based on that, we treat it with all those targeted agents, right? So this phenomena, this kind of approach is applied for many diseases, not only lung cancer, all of this disease. This basically showing that the new cancer drug approval and or indication by the US FDA is every year now, over the last 10 years, about 10 to 15 new drugs being approved by the FDA. And now last 1 year alone, about 32 compound has been approved. All days, I work in this area about 30 years, either few or even zero developed. Because of this molecular biology development, new drug development, this area not only in fact is cancer, but many other diseases also rapidly evolving right now. This is a great time. You guys in involved in this drug development. This is never great time. So this is you don't need to read this, okay? Too small. But a lot of the new drugs, either MAPs or NIPs, small molecular antibodies, and this is a disease mechanism, and these are also different companies here. Majority of the United States companies, Europeans, Japanese. Unfortunately, I don't see the Korean companies yet here. But that's what's happening here. So this is all of these drugs is been rapidly, I think this year, 2016, could perhaps even more than that, okay? So this slide I just purposely showing here, metastatic melanoma. The old days, dacarbazine is one of the drugs been approved for using this for several decades, but this is a nasty drug. Nausea, vomiting, very severe. And then high-dose interleukin-2 was developed in mid-1990s. That's also quite significant side effect, have a small effect. But recently we have a lot of molecular target agents. Ipilimumab, probably you know that BRAF mutation target. Nivolumab, the PD-1, PD-L1 targets. And this one is amazingly different. If you look at this survival curve, this dacarbazine versus nivolumab, huge difference, right? These are all metastatic melanoma, fatal disease. And this overall survival, now even more. If you example like ipilimumab plus nivolumab, is survival curve even better, like here. So this is huge. I think everybody knows this gentleman, right? President Coulter. He actually, this is already published, so I can tell. And this gentleman diagnosed of metastatic melanoma to the brain and liver metastases. Already resected and giving nivolumab, and he's doing fine still. In just recent press conference, he said, I benefit from better detection technology and treatment that didn't exist only a few years ago. So I think it's this is really clearly, we're showing that there's a lot of patients get benefit from this new discovery, new drugs. The reason I'm showing this one, this gentleman basically saying in the State of the Union address in January 2015, talk about precision medicine initiative. Know that this is a personalized approach, right? He's saying that doctors have always recognized that every patient is unique, and doctors have always tried to tailor their treatment as best they can to individuals. What if matching a cancer cure to our genetic code, keyword is here, genetic signature or code, was just as easy, just as standard. So based on this initiative, NCI embarked so-called MATCH trial, which is molecular analysis for therapy choice, that's MATCH. And this one launched August 2015, last year, after that State Union address by President Obama. Basically, you got the tumor tissue, sequence, and then you know which one is somewhat mutated or altered, and then you find the targets, and then treat and it's match. That's what you hear. Since August 2015, first patient entered throughout the country. Emory is one of them, Hopkins, or many institutions throughout the country. And then this one, within 3 months, rapidly accrued the patients up to almost 800 patients, very rapidly. Then this one actually presented in the ASL meeting in May this year. Now that treatment arm expanded 24 treatment. Initially was start with 10, now it's 24 different compound. And then increased the sample size of 5,000 all over the country, and then still ongoing at the moment. I'm sure it's going to very quickly finish that. And this is the 24 different treatment targets and drugs. All right, it's like here, EGFR mutation, afatinib, example, ALK translocations, crizotinib, on and on here, okay? So this is a really, really exciting time as an oncologist, as a drug development, companies like you. Another important program has been announced recently, so-called the Moonshot program. This is based on the Vice President Joe Biden's plea. Probably many of you know that his only son passed away because of this brain tumor, GBM. And then they actually initiated, we tried to get rid of this cancer from this world. And then they actually talking about in charge of mission control, which is, the 1960s, the first human beings landed to the moon, right? Through the Apollo plan. And then basically that's the same concept. Let's say we just launch and then get rid of this disease. So their side set of money is quite large, $1 billion just for this program. And then this program is going to be soon announced which disease and which application we're going to do and that things. Actually, Joe Biden in the April ASL meetings and ASCO meeting in June, he just came and talked about these moonshot programs. So what I like to say something about this drug delivery development, very complex signaling and precisions. This one is still a long way to go. Development drug resistance, this is another big issue, and also improved delivery of drug. Sometimes like a water soluble, not soluble becomes soluble, and then this kind of issues is like a paclitaxel example, and or non-specific distribution and targets in human body. So when you give the chemotherapy example, it goes to cancer cell, but it also goes to normal cell, giving you a big toxicity. All right. So this issue has to be addressed. And low concentration drug inside the cells. Current PK concept, pharmacokinetic concept, is talking about just circulation in the blood, but now through the new technology such as nanotechnology, we can address that issues. We can measure the inside the cell, the drug concentration. So clearly we need better technology. So I'm going to go talk about a little switch gear about nanotechnology. So I'm going to give you very quickly, because already he has the time over there, but the nanotherapeutics is a smart delivery anti-cancer drug by technology. NCI is actually defined as a science, engineering, technology conducted at the nanoscale, which is about 1 to 100 nanometers. So this is the size of the nano. So we're talking about here. Glucose is 1 nanometer, antibody is 10 nanometer, a virus is 100 nanometers, cancer cell about 10,000 nanometers here. In fact, one cancer cell can accommodate more than 100,000 these nanoparticles. So this is a major interesting delivery systems. Polymers, liposomes, cyclodextrins, it's all different now, carbon nanotube, gold, and DNA recently worked these nanoparticles for drug delivery. And it can be applied all different area. Okay, so I'm not going to go through that. So basic concept is this nanoparticle, there's a two-way active delivery and passive delivery. I'm not talking about passive. The active is basically this nanoparticle with a ligand and receptor conjugated, and this one can go to cancer cell if they have a lot of receptor on the cell surface. Normal cell may not have that. Then this nanoparticle go to only or majority go to cancer cell, not normal cells. Okay, so this kind of concept. In fact, we reviewed heavily about this one over the last 10 years. So there's many ligands we can talk about, but I'm going to give you 3 quick: the folic acid, transferrin, and EGFR single chain, and can target their receptors. So this is one of the drugs we formulated, folate and heparin, the Taxol, and then this is a kind of structure here, and you have size about 50 or 60 nanometers, which is an ideal size. And this is a preclinical study, colony formation assay, control and then Taxol treated. Some cells decrease, but nanoparticle with specific ligand, it's almost to kill the cells completely. And also, we did animal studies. This is Taxol alone. A lot of tumors still there. This is around here, and nanoparticles clearly clear up all the tumors. Of course, it's not been, this is all preclinical study, but if you this one achieve in the clinic, that's going to be fantastic, right? We haven't done that yet. And also, siRNA delivery is very important. You have all different ways. This is a very vulnerable kind of things, and when you circulate it and degrade it very quickly. So we have to safely deliver. This is a mRNA shutting down. Therefore, bad protein couldn't produce, right? So this is a concept. And basically, we start with RM2, which is very important gene transfer from ribonucleotide, the deoxyribonucleotide, and then this one shutting down its siRNA, all right? So the well-known investigator, scientist, Dr. Mark Davis from Caltech, actually he formulated this nanoparticle, cyclodextrin with adamantine pegylated, and then also conjugate with transferrin ligand and making it like a flying saucer, right? This one, in fact, did a phase 1 clinical trial conducted at the City Hope Hospital in LA. And basically giving this drug to the patients, and then if you look at the biopsy before and the treatment, nicely decreased mRNA expression before and after treatment of those patients sampled. These patients are almost all melanoma patients, which is very fatal again. And then also protein expression is decreased as well. So this is showing that probably you cannot show very well, but there's like a green lights that's actually infused nanoparticle you can actually showing through the fluorescence. And then this one, basically, those siRNA going to the tumor, and then you cut this one to defunctionalize this bad protein, bad messenger RNA. And then this is basically showing the 200 base pair. This is a control cell, and the sequence exactly the same they can find. Actually, this paper is also published in Nature 2010. With collaboration in that group, we actually did this color 1 is nanoparticle name. And then if you see that, this treatment, animals, and all controls, animal nicely growing this tumor, and then to treat it with nanoparticles, clearly decrease nicely tumors. In 2010, NCI embarked so-called nanotechnology alliance throughout the country, East Coast, West, Midwest, Southwest, and California, and about 50 institutions participated. We are one of them too. This one, we actually so-called the CNPP, Cancer Nanotechnology Platform Program, and is using gold nanoparticle with photothermal therapy for cancer. And then this gentleman outside is a PI, the Georgia Tech site. I'm the PI of the Emory site. And basically here, this gold nanoparticle is high conversion energy to hit. So when you hit this one to the tumor after injection, these nanoparticles, cancer kills are highly effective. This is one of the preliminary data. It's showing here all the control, all tumor nicely grow, but nanoparticle with the laser is a flat out. The tumor doesn't grow, even one single treatment. So this is a very highly exciting stuff. Then it doesn't show very well, but these are DNA nanoparticles. This is another building blocks of DNA. DNA is one of the gene structure, right? But this one actually we are making as a carrier. And this one we call DNA origami, which is initially the Dr. Rothman published in Nature 2006. making all DNA like this kind of different structures. The further expanded library by this young man, Dr. K. He was working at MIT, and currently he's junior faculty in Emory right now, so we work together. And basically, these nanoparticles have different size. We can conjugate it with siRNA or even small molecules. We can conjugate it and let it go to specifically the tumors. So this is one of the preliminary data, and then actually the conjugated nanoparticle go into the cells as time goes here. And also we did a EM study, and nicely this nanoparticle go into the cell. Actually, DNA couldn't see by the TEM or electron microscope. So actually, we are tagging gold nano and then let it show it here. So this is very nice demonstration. Still, this is unpublished data. So one of the comprehensive program we are going into, or has been now, is the so-called SPOR program, Specialized Program of Research Excellence. It's the over more than 40 faculties involved. This project, I'm the overall PI. We have 4 major projects, 3 cores, and also development program, which is for the junior faculty. And then we have quite a good amount of funding from the NCI. So I think we don't have any much time now, so I'm going to wrap it up here. So many drugs cannot reach the clinic because of the complexity of the drug development. All drug we call Death Valley here. So if you have one drug approved, we start with a 10,000 compound screening, and then through the preclinical and clinical studies and get the approval. This is a huge, difficult, complex pathway. But nevertheless, we have more and more quickly, we're moving on that. So I just want to mention the one word, the clinical trials. Safety is the most important thing, and efficacy. Therefore, all the clinical trials must do with a very rigorous way, okay? So there's no exception. If you have these problems and safety issues later, you're in big trouble. So you have to be very from the phase 1 trial. And then future directions, we are in the best time for new drug development, no questions. Molecular-driven discovery to be continued, develop the drugs in personalized medicine or precision medicine based on molecular characteristics of each individual patient. And then nanotechnology, I believe, is going to be a highly promising field in biomedical engineering and science. It may open new chapter of therapy, including cancers and other diseases. And finally, academia, industry, government, regulatory agencies must work together for effective and timely development of these new drugs. So I think one of the examples, I'm talking about small business innovative research. This is NIH funding, small, and also a small business technology transfer program. This is one of the NIH program, and actually set aside this year, I believe about $870 million set aside for this one. This is still quite small, but this is going to be expanded more and more. In Georgia State, we have also called Georgia as a state level, very similar programs. I think KIT, and I believe you have some similar programs initiated. That's, I really congratulate such an effort. So drug discovery development is a team science, including many special expertise, medicinal chemist, cancer biologist, animal scientist, biomedical engineer, industries like you, regulatory specialist like FDA, KFDA, clinical investigators, work together. All has to be worked together. I think I'd like to wrap it up here. I have a lot of people works in my program and also collaborator in the Emory University, and also many people in Georgia Tech, MD Anderson folks, and also NIH funding. Thank you very much.